What's that? Yeah, that's your axon terminal, exactly, right. So the axon terminal, and that should be, I'll go ahead and draw this in black too, but junction up with, now we're talking about a neuromuscular junction, maybe we should make it a little more wavy, right? And then we gotta draw something. Don't need that later. It's right? So what is this other side of the neuromuscular junction? What's this whole thing? The black line that I just drew, except for that. So yeah, this part here, right, motor end plate, which is part of the larger membrane of the muscle cell called the what? Yeah, the sarcolemma. I'll label that. That's your sarcolemma. That's your axon terminal. And then, okay, so we got a couple of key structures in place. So let's see what is follow the signal along, right? Maybe I got 3 colors, so we can draw that in purple, right? So symbolized by these little kind of arrows that go along membranes, right? What do you call that? What is the signal that leads to a muscle contraction? Yeah, in the nerve right now, it's an action potential in the neuron, right? So this action potential gets down to the synapse, which is all of that, a neuromuscular junction. And then I told you about this step in the muscle where you have some calcium going on here, but we didn't talk about it for the neuromuscular junction. I mean, what actually does happen is that there's these voltage-gated calcium channels, right? Line draw there, and that when that action potential reaches those voltage-gated calcium channels, that lets in calcium. Okay. And that's the calcium is, I mean, it's an ion, but it's not really used directly for its charge. Instead, what that calcium is going to do, it's going to cause the release of these vesicles, which that's a structural component too. So I'll draw them in. Draw the black. They're all throughout the axon terminal. Some of them are already docked with the membrane, and they're like ready to go. So when that calcium enters, they trigger the release of these vesicles into the synaptic cleft. So in a neuromuscular junction, what is it that those vesicles contain? Acetylcholine, right? It's a neurotransmitter. This one in particular is acetylcholine. We're going to talk about the remainder of chapter 11. Most of what we have to cover is talking about neurotransmitters. So there's a bunch of other types, but in the neuromuscular junction, it's acetylcholine. Sure, there's some exceptions to that. So what do you need on the muscle side of things? Let's see, I drew. What do you need on the muscle side of things in order for acetylcholine to have any effect at all? What does acetylcholine have to do after it enters the synaptic cleft?
Sodium channels.
Yeah, exactly. It binds with ligand-gated or chemically gated, same thing, sodium channels. So acetylcholine, I just drew a little purple dots, I'll draw it there. Bind some channels, and we let in sodium. Well, that sodium is positively charged, right? So, you let in a bunch of sodiums over the, I'm trying to draw pluses, but I've got my marker ring drawn one. Here we go. So you make it more positive, right? That's known as what? Depolarization, right? Because the membrane had this charge across it. It was negative. I let in positive ions and made it less polar, depolarization, right? Okay. How do you get further down though? You don't want to just stay here, right? You got to get where all the muscle, myosin, all that stuff is, right? The sarcomeres, right? So how do you get from here elsewhere? I mean, you're going to propagate just like you do this action potential, just like you do in a neuron, right? So how do you do that in a neuron or a muscle? How do you get move along the membrane? Yes, voltage-gated channels, right? So these were ligand-gated channels here at the muscle end plate, right? Because in response to acetylcholine, they let sodium in, right? That's ligand-gated or chemically gated, right? But so there's this local depolarization, right? And that's going to open voltage-gated channels, which are going to let in the sodium, yeah? and so on and so forth. You know, they're all over the membrane. So when you open some of them here, you know, you can make the others reach their threshold and open some of them down there, and eventually spread all throughout the muscle in 3 dimensions. This is 2-dimensional drawing, but you know, muscle is 3 dimensional. They're all over the sarcolemma. Now, in order to really get to where, like, it's going to challenge, I'm really just talking about the electrical conduction right now, and that's all I necessarily expect you to be able to draw, because I think this has a lot of parallels with, this is a lot of similarities in how this happens in a neuron, right? In a neuron, the way you start the depolarization, well, the way you get like graded potentials in a neuron is through these ligand-gated channels. And if those are enough in a neuron, if those are enough to trip the voltage-gated channels in the axon, then you get an action potential, right? Now in a muscle, if this is enough to start the action potential, which hopefully it is, the action potential spreads all throughout the membrane, right, rather than an axon, because muscles don't have axons. But anyway, in order to really get into where the sarcomeres are, which I guess that's a structural component, you know, I'm just going to kind of draw them in the background because I'm not going to draw these whole sarcomeres, but these things are kind of, that's kind of supposed to represent a myofibril. Maybe I should write it to make it easier, right? Or, which myofibril is talking about both the myosin and the actin, right? And they're all over, I just drew one. But what are these infoldings of the sarcolemma that come into close contact with myofibrils and kind of go into the cell, although really they're open to the outside, you know? But what are they called?
T-tubule?
That's right. That's T-tubule. That's T-tubule. T for transverse, because they cut across the cells, you know. T-tubules, you know, and then in the T-tubules, let me scale back this myofibril so I can draw this a little bit more. Right, very close to the T-tubules, there's another structural component here. Okay. Maybe I should draw that in a different color, do you think? Sure. There's, you know, I don't think I'm going to do too much justice with the kind of branching pattern, so I'm going to make it real simple, but you know, you get the idea, right? Anybody tell me what I'm attempting to draw right now? Yeah, terminal cisterns of the sarcoplasmic reticulum, right? So that's important for continuing to signal on, because we got this action potential, and there's voltage-gated channels in the T-tubule, so it can travel into the T-tubule. But then, how do you get so the final step in making these myofibrils actually start contracting, or, you know, is releasing calcium. Okay, because calcium will bind with, what is that protein that calcium binds with to start muscle contraction? What's the T? Troponin, right? Yeah. Because once you bind the troponin, then the sarcomeres are free to do the whole contraction with the cross bridge formation, and that's a whole process. Not what I'm talking about right here, it's one you're going to notice in the slides. But how do you get the calcium into the cytoplasm, or the sarcoplasm, cytoplasm of the muscle? But where is it before the signal reaches this area? It's where? In the SR. You know, and exactly, and that's true for skeletal muscles. Although of course for smooth muscles, it's not really, because it's from the outside. But we're talking about skeletal muscles. So yeah, calcium is sequestered. It's concentrated. Didn't really leave a lot of room, but you can just imagine there's a bunch of calcium in the SR, right? In those tubules, and between the there's actually a couple of proteins. So I'm just going to kind of draw a couple of little blocks there. But probably more than a couple actually. But there are voltage-gated proteins right here that contact these calcium channels, right? The calcium channels are in the SR membrane. The voltage-gated proteins, or the voltage-tripped, I don't know if they're really gated because they're not technically channels themselves, but the voltage-sensitive proteins are in the T-tubules, right? The voltage-sensitive proteins, they sense the depolarization moving along the T-tubules, and then they open the calcium gates in the SR, because they're right next to each other. So one pulls on the other, pulls up the calcium gates. Then calcium can go out I didn't really leave enough room to draw. Like I said, I'm not that much of an artist here, but you can imagine at that point the calcium is going to exit the SR and be in the sarcoplasm, right? Where it can do its job of getting those troponins and tropomyosin out of the way. All right. So you get that. I mean, I might ask you to label, like, draw something similar to this. Maybe not this complicated or this many parts. Just show me, like, maybe one step, you know? Show me, you know, acetylcholine crossing the synaptic cleft and binding with stuff, and label a few key components of it, you know? Just for a few bonus points, you know, at the end if you've got time.